Everyone, welcome to the last and final session of Microscopy 101. So today's session is going to be focusing on image processing and image analysis for microscopy. And so before we get started, for those of you who are joining us for the first time today, just a quick introduction about BiPS, or the Biomedical Photonics Society. So we're essentially a chartered student organization, part of Georgia Tech. And BiPS hopes to serve as a platform for interdisciplinary research collaboration, exchange of ideas, and just engaging people in the optics and microscopy community. We're also really proud to say that we're supported as an official OSA student chapter, and we have OSA funding. So just a quick introduction about why we wanted to do this workshop. So this essentially is just a very introductory series of workshops to get people familiarized with basics of optics and microscopy. And we hope that the participants will gain a better understanding of basic concepts as well as currently used imaging systems and be able to use them to answer different types of questions in biology. And we were also hoping that this would be beneficial for people who are preparing for qualifying exams or people who are new to the field and are just curious to learn more. So in case you missed the previous two sessions, we have recorded them and they are going to be uploaded on our website as well as on YouTube. So be sure to check them out. So let's get started with today's session without any further ado. So just a quick breakdown of what we're going to be covering today. We'll be starting off with image quality assessment and then talk about some techniques for image enhancements. And then finally, we'll dig a little deeper into image analysis and segmentation. So let's get into today's topics. And we would start off with just thinking about what exactly a digital image is. So any image essentially is formed. You have an object and then you have an imaging system or your optics, which, as we talked about in the previous session, is essentially made up of a set of lenses and apertures or diaphragms in any microscope. And then you have a sensor. Typically, this can be a camera sensor that's capturing your image. At the sensor, the image can be in analog form, and then it's converted to a digital form, and everything that follows from that point on is basically in the realm of image processing. So these two are just two different representations of this same process. The important thing to remember is that the initial image that you form is analog, and then you have an analog to digital converter that converts this to a discrete set of pixels with discrete values forming a digital image. So ultimately, the digital image that you end up with is just merely a collection of pixels, and the values of the pixels basically indicate the intensity of the image at different locations. So now that we have a sense for what a digital image is, we need to review some concepts about image quality and how we can assess it. And so we'll start off by just refreshing our memories about electronic detectors that we learned about in the previous session. So any electronic detector pretty much works on the same principle. So you have photons that impinge upon any photosensitive material, and they are converted from photons to electrons by a process known as a photoelectric effect that, if you remember, we talked about in the very beginning of the first session. So this conversion process is essentially controlled by something known as the quantum efficiency, which is just the efficiency of the conversion of photons to electrons. Now, these electrons can either be recorded in the form of a voltage or a current, and this is still an analog signal. So then you have an ADC that converts this to a digital signal. And then depending on the nature of your ADC, you can have different number of steps or different bit depth, giving you different numbers of discrete or digital levels. Okay, so now let's get into some different metrics to assess image quality. So we start off by thinking about digital images and how they are sampled on a detector. So as you can see in this image, the image on the extreme left looks pretty blocky and pixelated. And then moving to the right, the image quality appears to be better. And the only difference here is that the pixels have become smaller, or you've increased the number of pixels because you're using smaller pixels to sample the same area. So the real question here is, how would you decide what a good pixel size is? And would you be able to improve the appearance of this image by just consistently adding more and more pixels? Or is this governed by some sort of a fundamental limit? So in order to answer this question, we essentially go back to looking at the resolution limit that we talked about last time. So this is essentially given by d, which is basically the smallest distance between any two resolvable features. And that we saw was given by a relation between the wavelength and the numerical aperture. So the question now becomes how to be able to sample this so that we can distinguish two objects accurately. And so this is given by an extremely fundamental and old theorem from the world of signal processing known as the Nyquist-Shannon sampling theorem. And this theorem basically tells us that we want to have at least 2 pixels to represent the smallest feature, which in this case will be D in our image. In practice, we want to have at least 2.5 or 3 pixels, maybe even more to do this, but the minimum we need is at least 2. And the reason why is, as you can see in this image below, we can see 2 points that are just resolved, and you can see that right at the center of the line joining these 2 points, there's sort of a dip in intensity. So now if we had only 2 pixels, we would just see the 2 bright spots right next to each other, and we won't be able to tell if that's just a larger spot or if they're 2 different spots. So ideally, you also want to have a third pixel which records a slightly lower intensity to actually be able to distinguish them. So now we have this fundamental limit that comes from our imaging system. And like last time we looked at relating the pixel size to the field of view, we can also look at relating the magnification and the pixel size in our system to this sampling requirement. So just as an example, if you consider any typical camera pixel size, this is about 6 microns. And so the effective pixel size, just like we talked about last time, is just whatever pixel size you have on your detector divided by the magnification of your system. And depending on the magnification, this effective size would vary. So a pretty typical resolution limit for a lot of systems is around 220 nanometers, where you might be using a pretty high NA objective, say about 1.4 NA, and maybe around 500 nanometers as your wavelength. So now, based on our resolution limit, we essentially want the effective pixel size to be less than 110 microns. So we see that the required magnification in order to be adequately sampling would be close to 54x. So we can easily use a 60x objective and be able to sample adequately. The other option would be to change your detector, but obviously it's easier to just increase the magnification in your system, because remember, you can always add other lenses in your system to increase the magnification, and it need not just be dependent on the objective. So now that we have a sense of what should be the ideal pixel size, let's see what happens if we don't adhere to this requirement and don't sample adequately. But before that, just a quick introduction or review of what spatial frequencies refer to. So on the left, you can see an image of 2 different square waves. You have one that has a higher frequency, or you just see that this light and dark pattern is varying faster. And with a lower frequency, you can see that the bands are wider and there's a slower variation in space. So instead of just a square wave, if we had a sinusoid in space, this is essentially what it would look like. And you can see that between the left and the right image, the frequency is essentially the same. It's just that the variation in the maximum and the minimum values, or the contrast in the image, is slightly different. So essentially, you can think of any 2D image as being composed of these different types of sinusoidal patterns in different orientations and being made up of different frequency components. So as you can see from the image on the right, this is an image of a bunch of flowers, and the low frequency information is basically just giving you very coarse details and very broad scale variations in intensities across the image. And as you look at finer features or higher frequency information, you start to see a lot more detail and a lot more quick variation in the features. For example, in the edges, there's quickly varying changes in intensity, whereas here a large region of the image is almost uniform. So this is essentially what the spatial frequencies in an image refer to. So now let's see what happens if we don't sample enough and how different spatial frequencies might be affected. So the result of insufficient sampling is essentially known as aliasing. And to get a sense, we can just look at this in 1D. And so this is basically the sinusoid in black is essentially the signal you're trying to sample. And you can see that you can have 3 different sinusoids emerging of different frequencies depending on how you sample. So if you sample at a much lower frequency than necessary, it can look like the one at the bottom. And as you increase the frequency, it'll get closer and closer to the actual sinusoid. But unless you're sampling over the Nyquist limit, you won't actually be able to represent the correct frequency accurately. So in a 2D image, something similar happens. Essentially, if you're not sampling high enough, you can see that this image of a zebra, it looks pretty weird, and there's a bunch of these checkerboard patterns, especially around the snout. So the high spatial frequency features aren't correctly represented, and they're incorrectly represented as some lower spatial frequency. So as you increase the sampling rate, in the sense that you're decreasing the pixel size, you can see that the quality of the image is improving. And finally, when you get to the rightmost image, you see that it looks pretty good. You can pretty much make out all the features really clearly. And so this would be somewhere where you're sampling above the Nyquist limit. And beyond this point, any further oversampling wouldn't really improve your image quality, since ultimately your resolution is decided by the system. And once again, just a reminder here that you always want to make sure that you have the right magnification to ensure that you're sampling accurately, because irrespective of how great a system you have and how good resolution your objective might afford you, if you don't sample enough, then you'll end up with really bad images at the end. Okay, so now we talked about a really important factor, that is just being able to sample and correctly represent the images. Now let's look at the possible sources of noise in the image and something known as the signal to noise ratio. So one of the most prominent sources of noise, and something that we absolutely can't avoid, is something called photon short noise. And then this just refers to the fact that light is in the form of a particle as photons, and there's some amount of randomness associated with these hitting the detector at any given time. So you don't have a continuous stream of photons that go at a constant rate, but rather you have some randomness in the number of photons hitting the detector at a given time. And we don't need to get into the statistics that this process follows, but essentially we can just remember that the noise scales with the number of photons and is related to the square root of n, where n is the number of incident photons. We also have another type of noise that's associated with the detector, and this is known as read noise. So this is basically just the error in reading out the charge from the detector element. So like we talked about previously, the photons are converted to electrons, and then this is read out as a voltage. So basically this is just associated with the conversion of the electrons to a voltage, or basically counting the number of electrons in each element of the detector. So the important thing about read noise is that it doesn't actually depend on how much light or how many incident photons you have. And this increases with the speed at which you're trying to use your camera. So if you're trying to read out data very quickly, you can imagine that this process would be less stable and there would be more noise associated with it. So now let's look at something called the signal to noise ratio, which is an important image quality metric. So this is basically defined as the power of the signal to the power of the noise. Now, in most cases, we don't really have a clear distinction between some signal image and a noise image. So essentially, we would take any image that we have and use its mean as the signal and use its standard deviation as the noise. So let's see how this can be quantified. So we can consider the signal to be proportional just to the input intensity or the number of photons n. And if we consider the fact that we're mainly limited by short noise, then the noise is proportional to the square root of n, and so the SNR is proportional to the square root of n. And this just shows that to have a higher signal to noise ratio, we essentially need to increase the number of photons. And the way to do this is essentially to just expose your detectors for a longer time. And because of this square root relation, we see that for a 2 times improvement in the SNR, for example, we would need a 4 times higher exposure time. So the really important thing to keep in mind here is that the photon short noise is something that's fundamentally limiting and is something that's given by nature, and there's nothing we can do about it. You can always make improvements to the detector to be able to overcome the read noise, but ultimately you're going to have to deal with the photon short noise. And so the image on the right here is basically just illustrating this trade-off between the SNR and the time it takes. So on the leftmost end, you have a long exposure time and you have good signal to noise ratio. You have a nice looking image. And as you decrease the exposure time, the image gets grainier and grainier. So ultimately, there's always a trade-off between your temporal resolution, or how fast you can go, and what kind of signal to noise ratio you can get in your image. So let's look at another very similar and often related factor, which is basically the image contrast. And you can define a similar metric known as the contrast to noise ratio. So the contrast is basically just the difference in the intensities, either between a target signal and the background, or just between any two regions of the image. So if you have a specific, say, cell you're trying to image, you can consider that your target and everything else surrounding it to be the background, for example. And very similar to the signal to noise, you can essentially define the contrast to noise ratio as this contrast, which is this difference of intensity, is divided again by the standard deviation over the entire image. And this is pretty visible in the images where you can see, oh, I'm sorry, the high and low should be interchanged, but the image on the left essentially has a lower contrast, and you can see that the difference in intensities is not as high, whereas the image on the right has a much higher contrast. So now that we have a sense of these image quality metrics, another thing that is important to look at is something that's known as the bit depth and the dynamic range of your detector. So the dynamic range essentially just refers to the number of different intensity levels that we can distinguish. Just to give you a sense of what this might be, this for the human eye is about 100. So the dynamic range is defined as the full well capacity divided by the read noise. Now, on the left is just the specification sheet for an Orca R2, which is a pretty typical scientific imaging camera. And like we talked about earlier, the read noise is not really dependent on the amount of light you have. So it's independent and it's given as some number of electrons. And the full well capacity essentially just refers to how many electrons an element of the detector can hold before it has to be read out and emptied. And so this ratio gives the dynamic range, which for this camera is about 3000. And this is like an optimistic estimate, but you immediately see that it's many factors of magnitude higher than what we can see with our human eye. So now that we have this dynamic range available to us, it's important to accurately be able to capture this digitally. And that's where the bit depth comes in, which basically just refers to the number of gray levels that the detector can record. And you want to have enough so that you can capture the full dynamic range. What this means is that if you have something that's a 2-bit signal, then you can only record black or white or 0 or 1. Whereas if you have something that's a 10-bit output, you can have 1024 different grayscale levels. And as you can see in these images, the 2-bit image, you can just distinguish a light gray and a dark gray. Whereas in the 8-bit image, you can see a lot more distinction in different colors, and it's much easier to make out features, and they don't look as blocky as this image. So it's important to remember to be able to use this full dynamic range, which means while you're imaging, you want to have the intensity values close to, say, for example, if you have a 10-bit camera, you want to be close to 1,000. You don't want to saturate in the sense that you don't want to have a lot of pixels reaching this maximum value, but you do want to be close to that so that your intensities are spread across this entire range and not just using a small range where many different intensities would be recorded as the same. Okay, so now that we talked about some metrics for image quality, what would make a good image? Since we're talking specifically about microscopy images, the most important thing is that the image is, of course, in focus and that you have limited or very few aberrations in your imaging system. And then you want to make sure you're correctly sampling to actually be able to record all the information that you have. You want to have a high signal to noise ratio because you want an image where you can clearly distinguish things and you don't want it to be corrupted by noise. And you want a detector with a high dynamic range, and you want to be able to use this while you're imaging. And finally, you want to have good contrast because you want to be able to distinguish between a target or a sample of interest and background surrounding it. So now that we have a sense of image quality and what is really a good image, let's talk a little bit about image enhancement. So image enhancement, as the name implies, is basically just improving an image. And this can be either in terms of trying to improve one of these quality metrics that we talked about, or it can just be that you're trying to emphasize certain features and bring out certain features to be able to do further processing, or use this as a pre-processing step for other more involved analysis methods. So the primary image enhancement technique is just background correction. So background here basically just refers to any intensity that could be due to an offset in your camera or the detector, or just any surrounding media or having some background fluorescence. Basically any intensity that's coming from a region that's not your target region of interest. So if you look at this image here, you essentially see some sort of sample that this is a single channel fluorescence image, and you see a dark background surrounding it. So if you look at the histogram, which basically indicates the number of pixels having these different ranges of intensities, you can see there's a very large number of pixels that have some lower intensity corresponding to the background, and then a much lower number of pixels corresponding to the target. So you can essentially use this histogram to sort of fit a curve to this region, or use some statistical measures such as the mode or the median to be able to figure out what the intensity of the background is. And then you can just reproduce a matrix of the same size to get the background intensity and then just do a simple subtraction. You could also just pick a region where you know you don't really have any sample of interest. For example, this corner could be, or this yellow highlighted region could be a pretty good example of background signal, and you can just average over that region and again generate a matrix with that same intensity and do a very simple subtraction to get a background corrected image. Another type of correction that's often done, which is also a type of background correction in a sense, is something called flat field correction. So this essentially works by acquiring two images other than the image with your sample. So you basically acquire a flat field image, which is an image that doesn't have any sample. You keep the illumination, the exposure, all of that exactly the same. And the purpose of this is that if you have any non-uniformity either in your illumination or in the way the pixels of the detector respond, you can correct for this. So you can see on the image on the left that the center is much brighter than the edges. And once you've corrected for this, you get a nice uniform flat field. And the dark field essentially is an image acquired when you are giving no input to the detector. So basically your shutter is closed, there's no light entering it, but you'll still have some small leakage current associated and you'll still get some small signal. And so this dark field image is just to correct for that. And so finally your true image would be the measured image minus this dark field term, which is basically some sort of an offset, and then divided by this flat field term that's just multiplying this variation of intensity across the field of view. So for a lot of microscopy applications, this dark field term isn't actually a big issue and doesn't contribute much. So you can simply just get a background flat field image and just divide that by a measured image to be able to get a flat field corrected image. And oftentimes, even if you don't actually acquire a separate flat field image, you can just use your existing image and perform different types of filtering and processing operations to generate what would be a pretty good approximation of this flat field image and then use that. So here, essentially, what we're trying to do is correct for non-uniformities in the background or in the illumination. And by doing this, we're essentially just improving the contrast in our image and improving the contrast to noise ratio. Okay, so now let's look at something else, which is extremely ubiquitous in image processing, which is basically just different types of filters. And though this concept might be pretty simple, you can accomplish a whole host of extremely varied processes and have very different kinds of outcomes just using simple linear filters. So a filter is basically just a small 2D matrix or a kernel, and that's convolved with an image to generate some sort of an output. So depending on the type of kernel you use, you can have very different kinds of outputs. So the process of convolution is illustrated in this GIF here. And say if you have a 3x3 convolution kernel, you'll essentially place this kernel at some location on the image, and then each pixel is multiplied with the value of each pixel is multiplied with the value of the kernel, and then you sum over the entire kernel, and then that becomes your output exactly as illustrated. And so this is essentially how the output of your filtering would look like for a linear filter. So this here is just an example of what's known as a mean or an average filter. And this is, of course, the simplest type of filter that we will most often talk about. And so this is known as a smoothing or a low-pass filter, essentially because it's passing the lower spatial frequencies and some of the higher spatial frequencies in the image are lost. So common ways to do this is to use this mean filter, which is basically just a box of all the same values. Or you can use a Gaussian filter, which is basically just a 2D Gaussian that's multiplying, convolving the image, sorry. So if you have an input, you can see that the box filter and the Gaussian filter, the outputs look pretty similar. What they're essentially doing is blurring out the edges and giving you a more smoothened version of the image. And this is just other examples with a mean filter and a Gaussian filter. So the important property of these filters is that essentially they can remove noise. And so in this example here, you can change the width of the Gaussian filter, for example, or the window size, or also the standard deviation of the Gaussian. All of these amount to essentially the same thing. And this would give you a more smoothened version of the image. But you see that the SNR is improving and there's less noise, but you also see that you're losing some of the edges and the ability to discriminate some of these smaller features like these lines here, for example. So this is essentially the purpose of using these smoothing or low-pass filters. It's an extremely simple operation to be able to remove noise in the image and still get a smooth-looking image at the end. Now, the opposite of these essentially is high-pass filters. And some of these examples are just given here. You have Sobel filters that can be either operating along a vertical or a horizontal direction. So high-pass filters essentially try to perform what is the digital equivalent of a derivative or a finite difference approximation. And we don't really need to get into this math much, and it's actually not really hard to understand, but essentially you're just approximating the derivative as a difference of two things, both in one. So this would be the first derivative, and similarly you can do this for the second derivative as well. So the Laplacian is essentially like a combination of the Sobel X and Y in 2D. And as you can see, already this high-pass filter tries to tease out sharp variations in intensity or what look like edges in the image. And this is precisely what's done in edge detection. So edges are any regions in the image where you just have a sharp variation in the intensity. And edge detection is really important because it's an important pre-processing step for a variety of different operations and a variety of different analysis techniques that might rely on this. And the filters that we talked about just previously are the most simple forms. You can have more complicated, more involved algorithms like canny edge detection, for example, but even these simple filters usually do a pretty good job. So let's also quickly talk about nonlinear filters. So the filters we talked about earlier were essentially performing a convolution operation. Whereas with nonlinear filters, we can essentially perform a different sort of operation that's not just a simple convolution. And there's many different types of these, and they can range from very simple to very involved. But we can just talk about a median filter. And so what it does is essentially, depending on the size of your median filter, you look at all the intensities in that window size, and essentially you sort them either in ascending or descending order, and then that pixel is just replaced by the median of the pixels in its neighborhood. So the reason why these filters are popular is because you can essentially reduce the amount of noise, so you're improving your SNR, but you're also still able to preserve edge features. So these are just examples of images with two different types of noise. The first one is just uniform Gaussian noise being added to the image. And you can see that the output from the median and the Gaussian filters look pretty similar. But in this case, it almost seems like the Gaussian image looks better. The median filter does preserve edges, but you can see here, for example, the earring of this lady is no longer visible. So you might lose out on some features with median filters because you're trying to preserve edges and you end up with sort of a blocky looking appearance. But median filters, for example, do a lot better with other types of noise, like this type of noise known as salt and pepper noise, where there's just a bunch of pixels that either don't respond or get saturated completely. And so you can see that with a Gaussian filter, there's not a significant difference in the output image, whereas with a median filter, the image looks significantly better compared to the input. So nonlinear filters, this is just a very small taste of what they can do. There's a whole field of research that's just dedicated to looking at different types of filters and optimizing them for different applications, but we don't really have time to get into that here. So finally, just shifting gears a little bit, just a quick introduction to what might happen in deconvolution microscopy or other types of computational processes in trying to recover or improve image quality. So essentially, you can think of a ground truth sample, which is just basically a bunch of 3D spheres in some volume, and this is known as our object. So what happens in an imaging system is essentially, again, a convolution process like we talked about with the filters. So you have this object essentially being convolved with our 3D point spread function that Dylan talked about in detail in the previous session. And so what you end up with is that all of these spheres essentially become stretched, and you have these hourglass-shaped volumes in the image that you're actually able to acquire. Now, in deconvolution microscopy, like we talked about last time, you're just acquiring different slices, but unlike confocal microscopy, you have no way to block out of focus light or restrict it to light from a single plane. So what this would look like projected in 2D is essentially something like this, where you have information from different planes just being projected. So in deconvolution microscopy, essentially, we have this image that we've acquired that's the result of this convolution. And what we're trying to do is just reverse this process. So having some information about this point spread function, we're trying to reverse this convolution process, or essentially solve an inverse problem to use these images and get to the object, given that we know something about the point spread function and we can apply different types of constraints. Now, there's many different ways to do this, and a lot of the methods are pretty involved. But the simplest method could be just some type of filter. And what this results in is a big improvement in image quality, because you can now eliminate all the out of focus light and get a clear, crisp image that's in focus. So this is just a very superficial overview. There's a lot of cool stuff happening, and I urge you to take a deeper look into this if you're more interested. But now we've covered just a few different ways to enhance image quality, and just some quick things to keep in mind is that essentially the purpose of any images, particularly in research, is to be able to convey some information. And it's not really important that they look very good. So it's not like we're trying to have really good looking images to share on social media or something. And it's important to be able to preserve quantitative information. So it's important to always document what type of processing you're doing and what exactly you're doing. So you want to use tools like MATLAB or ImageJ as opposed to using something like Photoshop. And yeah, documenting and knowing what you're using at each stage is extremely important, especially with quantitative images. For example, if you take a phase image that's supposed to represent quantitative values and blur it completely, then the phase values will probably no longer have any meaning. So that's something that's really important to keep in mind. And finally, just a really good rule to remember is to never manually generate any sort of data, and work with what you have. And there are limits to how much you can do to digitally quality, and there's also a lot of theoretical limits. So it's important to keep those in mind. So now I'm going to be handing over the floor to Paloma to tell us more about image segmentation and analysis.
All right. Well, yeah, you can go ahead and move to the next slide. So now that we've covered, how to get a good image and how to enhance our images in the best way possible, we are going to talk a little bit about image analysis and segmentation with the purpose of knowing how to interpret the images. I think that's kind of the goal that we have for this section. So let's just jump right in and begin with the basics, which is, what is segmentation? I feel like it's a word that many of you have heard, and it just means subdividing an image into its constituent regions or objects. Of course, an image can be segmented in many ways. For example, this image that we have right here is segmented as each individual person, each individual car. They each have a value or a color. You could also just segment it by separating people, roads, and cars. There's a lot of different ways that you can segment it, and it really just depends on your goal. But when is this useful? When we're talking about microscopy, why would this be useful for microscopy? Well, here's some examples. Cell counting, so flow cytometry, you use segmentation and classification quite a bit. For tracking, so here to the right, we have in my opinion, really cool image of some neurons, and this is a 3D segmentation of them, where we see that segmenting these neurons and their axons and dendrites help us a lot to be able to track all of these connections. So that's really, really useful. Segmentation is also used a lot, even more recently, for tumor detection and applications like that. And there's of course a lot more applications. And all of the, it's very exciting nowadays to do something called automatic segmentation. So why would we do automatic segmentation? Well, machines, one, can sometimes see things that we may miss. For example, in this image, it would be really hard for me if we just had a grayscale image of these neurons to follow all of these axons. And machines can really see that way easier than I can. But also, machines don't get tired. So for the example of cell counting, a human's not going to sit down and count millions of cells to get some statistics about your blood. A machine can do a way better job than a human. So that's when we rely on machines and automatic segmentation. It is really useful. So in order to talk a little bit about segmentation, we will cover some of the basics, and we will begin by talking about thresholding. The goal of thresholding, or at least the initial basic goal, is to binarize an image based on its contents. So for example, if I take this image of blood cells and I make it grayscale, so now it's grayscale, look at that magic, which means we jumped from a 3-channel image, right, because colors are R, G, and B. As I was saying, we have this grayscale image, I'm so sorry y'all, and I can do a very, very simple operation, which means I can pick a value and say any value in this grayscale image above the value I pick is going to be a 1, and anything below it is going to be a 0. If I do that with a value such as 230 for this specific image, I can binarize the image really, really easily. But say instead of getting just the background versus the foreground, say I wanted to only threshold the nuclei of these white blood cells. Well, I can do the same thing by applying a different threshold that is lower or higher depending on what I'm trying to keep, and very easily just get the nuclei. So how do we choose these thresholds? Because it looks like I just picked a number in this case, and I did. So you can do this manually. You can do something called manual thresholding, and you do it to separate known regions, specific things that you're looking for. A good example on when to do this is when you have phase values, for example. So if you're doing quantitative phase imaging and you know the height of your sample that you're trying to segment, you're able to pick that, a threshold that matches your expectations for what you're trying to keep. The same thing happens when you're trying to segment based on color. So for this image right here of the peppers, I want to keep only the red peppers. Well, that's a very simple segmentation when I know that I want to keep only a specific color. Now, when we're doing automatic segmentation for medical image processing, we will usually rely on automatic thresholding. And the most common method for this is the Otsu thresholding technique. This is based on the histogram of the image, and it is also based on the assumption that most natural images have a histogram that looks kind of like a binomial distribution, right? So we have 2 lumps essentially, and if we're able to separate those lumps, we can find the ideal threshold that would give us the background to the left, that first lump, and the foreground to the right, that one that will turn orange once this video is over. This would be the simplest way to the threshold, and there's obviously other techniques for non-natural images, but we usually rely on Otsu. There are also multi-thresholding techniques that rely on Otsu, and that means that you can break an image into different steps of thresholds. And we were not going to go into that, but just know that those exist and that you can also do that. Now, thresholding is cool, but it's often just not enough, right? So when I was trying to segment these cells, I realized that by just thresholding, I also segmented the platelets in the blood, which are not the cells that I'm interested in. So I was not interested in the platelets, and I wanted to get rid of them. So what do? Well, I did a very simple binary operation, and here's the final image that I got. The same segmentation, but now I got rid of all the tiny dots that are the platelets that got segmented as well. Paloma, how did you do that? Well, I'm glad you asked, because I'm about to tell you. Well, most operations that are, these simple operations for binary images rely on these 2 main operations. One is the erosion, and the other one is dilation. So how do they work? Well, first you have a structuring element here to the left. The green cross would be our structuring element for this example, and a binary image. What you do is you slide the structuring element through the binary image, and then you have 3 possible scenarios. The first scenario is that all the values that align with the structuring element, so with the green parts of this matrix to the left, are zeros. If all of them are zeros, in the erosion operation, you will set the center pixel as a 0. In the dilation operation, you will also make the center pixel a 0. Now, if they're all 1s, in erosion, you set everything as a 1. In dilation, you also set everything as a 1. Now, they seem pretty similar, but here is where things change. If there's any zeros in the erosion situation, you set the center pixel as 0. Now, for dilation, if there's any 1s, you set the center pixel as 1. So you may be noticing that erosion is driven by zeros, whereas dilation is driven by 1s, which means that when you dilate an image, anything that's a 1 will always remain as a 1, but you may change zeros to 1s. With erosion, it's backwards. Everything that's a 0 will remain as a 0, but you may change 1s to 0. So let's walk quickly through a very simple example that we've created. We have this binary image to the left and the structuring element. The structuring element laid where it is right now. Now we're seeing that there's any zeros. Are there any zeros? Yes, there's 2 zeros, which means that that central pixel that was a 1 before will now turn to be a 0. Now for dilation, in the same spot, are there any 1s? Yes, so it will remain as a 1. Let's look at a different position. Now we have it over there. Are there any zeros? Yes, so we keep it as a 0. For dilation, are there any 1s? Yes, so we change that 0 to a 1. And here are the 2 specific examples. If there's all 1s, if we keep the value as a 1, and for dilation, welcome to the world of online life. I hope everybody was able to follow, though, how we got those. So now let's look at the final results. For erosion, this is the final result of eroding this matrix with that structuring element, and that's what it would look like for dilation. Just know that the structuring element can change, and you can choose it. This is just the one we chose for the example. Now, there's 2 operations that rely on erosion and dilation that we use very often. One of them is an opening operation, in which you first do erosion and then do dilation, and this is the result. The other one would be a closing operation, in which you first do dilation and then erosion, and this is the result. So for the previous image that I showed you, when I got rid of the platelets, what I did was an opening operation. I got rid of the smaller pieces or segmented parts of the image while keeping the correct size, which was really important, of the other cells that I segmented. And that's the goal of using opening and closing operations rather than erosion and dilation. You want to keep the correct size of your segmentation. There are other operations, like for example, some people use filling operations quite often, which means any area that is surrounded by 1s will be converted to 1s. And MATLAB has a great library of binary operations that you can use, but just know that a lot of them rely on erosion and dilation. So now that we've talked about grayscale thresholding, we need to discuss the fact that color plays a huge role in segmentation for a lot of the microscopy images. So for example, if you're doing bright field, like that other image that I showed you, you have color images. You can use the color of the images for segmentation. If you're doing fluorescence, you also rely on color quite a bit. So let's look at the other image that I had before with cells. You can break it into different kinds of maps, right? So the color encoding that we chose for right now is RGB. The channels from left to right are red, green, and blue. And this is what those channels look like. You can already tell that, for example, with the green channel, you can really separate the cells versus the background. And you don't really need the other channels that much. What if we looked at the same image in a different color encoding method? So what if we looked at HSV? HSV stands for hue, color, saturation, and value, which means brightness, right? So we can play with these channels and get different results that we did when we were working with the RGB. Knowing that there's different channels and how to use them can be very useful for segmentation. So here's an example. You know that I segmented that image into all the different cells, including the platelets, right? I did so by looking into just the green channel. So if you look at the RGB thresholding, you can see that only by moving around the green channel, you can segment this image. If we looked at it from the HSV perspective, you will be able to tell that I actually need to manipulate 2 channels. I would need to manipulate the hue, and I would need to manipulate the saturation. So for this specific example, using the RGB channels would be a better choice. Now, say I wanted to segment the nuclei like I did earlier, right? So when we segment the nuclei, if we look at the RGB segmentation that I would have to do, I would have to apply thresholds to all 3 channels. But if I did it by only looking at the HSV, I could uniquely use the saturation channel and get a wonderful mask of just the nuclei. So that's extremely helpful. So far we have covered any sort of segmentation, relying on the brightness or the intensities of the images, right? So you can do so with grayscale, or you can do so with color segmentation. But sometimes, quite often, looking at just color or intensity differences is not enough. For example, I do quantitative phase imaging, and we rely on a grayscale image, but it's hard to, tell the foreground from the background. A lot of the times, the whole image is tissue, and the only way to differentiate between different sorts of tissue is by looking at texture of the tissue. So let's talk a little bit about texture analysis and some basic concepts, why it's useful. Here's a very, very simple example. A lot of people use this image to talk about texture. You can really tell by looking at this that there's 2 clear textures shown in this image, right? How can we tell our computer to recognize that? Well, there's a lot of different features that you can extract to do so, but here's 3 very simple ones. First, we have the entropy. The entropy essentially measures how disorganized an image is. So you can tell the clear differences over here from like a white noise of high entropy to just really straight lines, which would have lower entropy. We also have the standard deviation. I'm assuming you all have heard that, but it just means how separate that the distribution is. So a smoother area would have a lower standard deviation, and an area that is more granular would probably have a higher standard deviation. Similarly, we have the range. Sometimes the range and the standard deviation give us similar results, but sometimes they give us different results. It really depends on the distribution. So it's interesting to have them both. These are very simple distribution-based features that we can extract. So how would we apply these for segmentation? Well, using this exact same image, if we did the standard deviation and we applied a standard deviation to a small window and just slit the window through the image, so we would do a sliding window, we can get something like this. If we did the entropy, this would be the result. And if we did the range, this would be the result. I know that here it's kind of hard to tell how we would use these, so I think it's better to show an example of something that y'all are now very used to looking at, which is that cell image that we had before. So if I applied standard deviation, entropy, and range filters to this with a moving window, just like I did with the other images, this is what we get. You can tell already that the standard deviation and range, 1, look pretty similar, and 2 are doing a pretty interesting job at, selecting the edges of the image, whereas the entropy is just really segmenting different regions. Now, I can play around and change the size of the window that I use for this segmentation. And if I make it larger, now we get really different and interesting results. I may want to use the range to help me extract the whole white blood cells, which was a really hard thing to extract just by looking at the intensity of the image. And I can tell you it was hard because I tried, and it's actually kind of hard. So combining some of these filters may help us get really good results when we're trying to segment these. So just so you know, these are the most simple, some of the most simple texture analysis features that you can extract. And these are based on the histogram really, really simply. There are way more advanced features that may extract more hidden, quote unquote, hidden textures. And there are such as like the fractal analysis, which can tell you about repeating regions of an image. We can extract features related to the co-occurrence matrix, which essentially analyze the relationship between different pixels in the image, depending on their distance. So there's quite a few different ways to extract texture from images, but this is a very simple one, just so that you can see that it is important. So now that we know how to segment things in a very simple way, we may have realized that sometimes segmenting an image, for example, the cytoplasm of these white blood cells, is a lot harder than we thought just by applying thresholds or even applying, some basic texture analysis. And sometimes, not always, but sometimes we want to rely on machines to take care of the more advanced texture, segmentation or classification problems that we may have. So I want to talk about this very briefly. I will not dwell on it because, this is a huge topic, but just as an idea of what machine learning is and how we use it, well, for machine learning, if you had an image, the first thing you need to do is think what features could be useful to extract from this image. So then you extract those features. Once you extract those features that are useful, you may want to choose some sort of classifier that you can feed the features into and train it to optimize it. And then you teach a machine to do all of it. So then you would create an algorithm that extracts those features and pushes them through a classifier. There's a different kind of automatic classification or segmentation technique, which is called deep learning. And I'm sure you all have heard of it. And it almost removes the human from the scenario, right? So what you have now is a bunch of images, and you throw the images into the machine and tell it, okay, now you learn, figure it out, learn your stuff. While deep learning is, a lot more complicated, meaning you need a lot more data, a lot more images, and it takes quite some time, I would say a lot longer, to train a deep learning algorithm like a neural network, you can solve way more advanced problems. So before with the machine learning algorithm, we were throwing a cat image in, and we were asking the machine learning algorithm, is this a cat or is this not a cat? Now this deep learning algorithm can tell us whether it's a cat or not a cat, but it can also tell us what kind of cat this is, which sounds like a very complicated problem, but it is solvable if you have enough data. So this is kind of like the very, very basics of what these automatic segmentation and classification tools do. Now, when you are not very used to using machine learning and deep learning algorithms, you may feel like, everybody's using this and they're so useful and they solve all of these problems, but I do want to warn you to never trust them too much. We feel like we can rely on machine learning and deep learning algorithms a lot, but in this class we're talking about optical microscopy applied to the field of biomedicine. And in the field of medicine, you cannot rely on anything, not even humans, 100%. Of course, having more tools is always helpful, but you can't always rely on them, right? So for example, this is a really interesting Nature article that shows that if you feed a neural network algorithm a photo of a panda that would usually be classified as a panda, and just add white noise, a combination of colorful white noise to it, it can make the completely wrong decision. So we've talked about how microscopy techniques can, add noise to our images or add known aberrations to our images. If you feed a neural network algorithm an image with noise that was not expecting, it will, in fact, get confused. Also, neural networks or machine learning algorithms do not know how to deal with new information very well. So if I've trained a neural network and I've told it to tell me whether what I'm giving it is, dogs and cats, and then I throw a snake in there, the neural network algorithm will tell me whether that is a dog or a cat, but that is neither, and it doesn't really know how to say neither. Whereas the human brain knows how to identify things that are new, maybe not say what they are just yet, we need to figure it out, but we know how to identify something that is nothing we've seen before. And that is a very important tool that we have in our own brains that a neural network will not be able to solve. So what I would usually say about this is that generally we want to rely on neural networks and machine learning algorithms as guidance rather than like final decision making techniques, uniquely except to when the problem is really not solvable by humans. So what I mentioned before about cell counting, right? If there's going to be millions of cells, humans can't really do the job that a machine learning algorithm will do when it comes to classifying cells in flow cytometry. So we will rely on a machine learning algorithm, and that's okay, or some sort of classification, that's okay. But generally, say for tumor detection, we may want to use it as guidance, but we really want the doctor to take a look and make sure that the machine is making the right decision. So just keep that in mind. And then there's one more topic that I want to talk about today that is kind of unrelated to this, but I think it's important to mention, because if you will be working with microscopy, you will run into a time where you need stitching to happen. We've talked about, how our different microscopy systems will have a field of view, for example, that we can acquire. If I have a sample that is 3 by 3 millimeters large, and my system can only acquire 500 microns of as a field of view, I need to take multiple images to be able to get the whole sample that I'm trying to image. And then I will have to stitch these images. Thankfully, nowadays, we have very advanced stitching techniques that are completely automatic, and we don't have to worry about it. But I do think it's important to at least kind of know how they work to be able to move forward. So there's 2 steps that these techniques use. One is to figure out the overlap between the images, right? So how do we do that automatically with the high precision that microscopy requires? You want every pixel to overlap perfectly. Well, a very, very simple way to do it is to use the cross correlation of the 2 images. Here's an example of the reference image that you would have, and then you have an image that is offset a little bit to the top, like moved up a little bit. If we do the cross correlation of these 2 images, the maximum value of this cross correlation is translated a little bit. It's moved a little bit from the center, and that will give us the actual difference offset between these 2 images. So that's a very simple solution, but there's way more advanced ways to do registration and to do, to find these overlaps. So for more natural images, like when you're trying to take a panorama image with your phone, it is very common that you will not only move, but maybe rotate your angle a little bit, get a shear in there, maybe there's a change in magnification. The best way to do this is to extract different features from the images that you take from both images, and then try to overlap the features well, not only the pixels, but the features. So for example, what's happening here is that some of these dots may be different edge directions of the image, or different like color jumps and things like that, and that's how your phone can stitch these images. Now, how do we stitch them seamlessly, like we see all the time? Right? How do we get from the image in the left to the image in the right? Well, there's many things that you can do. The first one is to make sure your background is fully flat, which is something that Nish was talking about. Get rid of, do all of the background corrections you need. And then there's a lot of fusion techniques that you can use to improve the merging of these images. Now, there's a ton of different solutions. I don't think you are going to care about the details. I think what you really need to know is that there's a program called ImageJ. And you should rely on ImageJ for stitching unless your microscopy system already has a stitching algorithm, which is great. Some microscopy systems, when they do scanning, they also do their own stitching. And you can fully rely on that. Or if not, you can take your images to ImageJ and use this. I personally prefer the linear blending fusion method, but you can play around with them. And there's a lot of different techniques for that. So now that we know all of this, we've essentially completed.
Thankfully, this is essentially our last topic for today. So finally, just want to plug some resources that y'all can look into. We have a bunch of courses listed here that you can take if you're interested. These are really good classes that are super useful. There's also a bunch of online resources and other resources. We'll have a much more detailed list available on our website. But yeah, these are definitely things you should look into. So now I'm just going to hand over the floor to Dylan to deal with our question and answer session.
All right, thank you for following my niche. Once again, we appreciate you all for joining our virtual series of Microscopy 101. All content will be uploaded in our homepage. So let's go to next page, please.
Sure.
So yes, so any Q&A you have, please feel free to go ahead. You can hands up button on the right side panel or chat, or feel free to unmute yourself for questions. Thank you.